Yeah, hello everyone. I hope you can hear me. Someone please acknowledge if you can hear my audio. Yes, yeah. Thank you, Deepika. I hope you guys have gone through the slides for this week. If you guys have any doubts from the lecture sessions, you can ask me, or even after class, you can message and ask. And even if there were any questions or doubt from the previous session as well, you can still ask. I'm gonna just wait for another 3 minutes so that I'll share my screen and start. People are just joining. I hope all of you are solving your assignment questions, the assignments that are provided along with the course. If there are any questions from those assignment, you can ask. We can solve those questions together. Okay. Uh, Prarthana has a question. In the given assignment of week 3, question number 6 is while using mathematical modeling for integrating a large number of microbial community of environmental significance, which is the most essential system component that must be identified by a mathematician? Which one of this is correct? Critical type of microorganism, substrate consumed, utilized by microorganisms, product formed by microorganisms. Uh, we will briefly look at this today. And if you still have the same question after we finish the session, we can again come back to this question. Thank you for acknowledging. Just one more minute. If anyone else has any questions, please do post it here. Okay, it's 6:05, so I'm gonna start sharing my screen and keep accepting people as they join. Let me just do this. Is my screen visible? Could someone please unmute and confirm? Is it visible? I can't see if you type it in the comment box, so please confirm if the screen is still visible.
Yes, ma'am.
Yeah, okay, thank you so much for confirming. Yeah, let me just do this again. Yeah, so this is session 3. Last week there was a lot of disturbances in the middle of the session. So I had to upload the video as 2 sessions. I hope you guys got the links, the YouTube links for both the sessions as well as the PowerPoint slides. In case if there are any questions from last week's session, you can still ask those as well. And I hope you guys were able to understand from the previous session. So today's section is going to be mostly an extension of what we looked at yesterday. Yeah, I mean, not yesterday, I mean in the last session. In the last session, we saw about microbial ecology, the different relationships that microbes have with themselves or other organisms or with the environment and the abiotic factors themselves. So now we're going to look at some of the applications of how to use some of these things when we understand how microbes interact. How do we use that for our own knowledge? So any country, any developing country or big countries, produce a lot of waste. This is mainly because of the population explosion and also because of the kind of lifestyle that we all lead. So we produce a lot of waste, and that waste, if it is a liquid waste, it ends up in the rivers polluting water bodies, or if it's a solid waste, it ends up in the landfills, producing polluting our land and environment, and it just stays there forever. We saw about biodegradable, non-biodegradable waste and all that. So any country, any developing country, before investing more on more technological advances, should invest more of their resources in maintaining a better environment, in trying to address how to treat the waste that is being generated in the environment. So any country would benefit from investing in environmental solutions. And the main resources that are affected, like I said, by the environment, or soil, water, and even air also gets polluted majorly because of the modern technological advancements that we have. So we'll see some of the applications of microbes. So earlier in the briefing class, or in the first session, we saw about Pseudomonas putida, the superbug, which was developed by Professor Anand Mohan Chakrabarti. And we briefly saw how it can help in controlling oil pollutions. It also has a broader range of stress tolerance. It can actually be used for clearing up oil spills of larger volumes, and it can tolerate a large range of stress, including temperature and pressure stress. It is helpful in bioremediation. It is a self factory, that is, it produces higher amounts of energy when it breaks down the oil resources. So it does not just break down the oil particles, it also produces energy when the breakdown happens. And it's an amazing strain for genetic manipulation. That is how the oil eating strain was also produced. In one of the classes, someone has asked a question about how do we mimic the conditions that are there in the wild for a microbe that we are growing in the lab or culturing in the lab. So this kind of culturing techniques are mostly trial and error based. These laboratory techniques for culturing microbial organisms that can perform bioremediation is broadly based on the fact that we can manipulate their genes to produce strains that require less of maintaining conditions and can perform our waste remediation of interest with limited resource availability. And that also includes stress tolerance, that is, they can grow under a wide range of conditions without requiring a particular substrate or a particular condition to grow in. Biopolymer accumulation. Okay. Yeah, I have been requesting people in the past three sessions, please do join before the first 5 minutes so that it's seamless experience for the rest of the people who join early. So please join in the first 5 minutes of the class. In case you can't join, also the recording, the full recording from the beginning to the end will be available on YouTube. So you can always go back to the YouTube recording and check the video in case you missed the lecture for the day. Yeah, so biopolymer accumulation, biofilm formation, these are some of the things that are again associated with utilizing the waste itself for production of secondary products that can be further used for other purposes. And root colonizer, these also live on plant roots, so they have so we saw about mutualistic relationships and symbiotic relationships that microbe have with plants, and we saw about the example of nitrogen fixing microbes and leguminous plants. So like that, putida can also be a root colonizer and help the plant. So these are some of the advantages of Pseudomonas putida. It has a very higher efficiency. It can uh, work under stress conditions with very high efficiency to chew up the oil molecules, the oil polymers. And it also has a spectrum which can work on a wide range of organic matter. It can work on very different types of carbon bonded structures, and therefore this makes the Pseudomonas putida a superbug. But understanding the ecology comes first, because only after that we will know what kind of substrates it can utilize, what are all the different products that it can produce once the substrate is utilized. A major application of microbes is treatment of wastewater. So from here, we will look briefly about how wastewater management can be done with the help of microbes. So this is an image of a conventional wastewater treatment process. So the existing activated sludge, so this is any wastewater sludge or sewage water that is produced in urban settings. So this kind of, okay, I will just, yeah, so this is the wastewater which then goes into this activated sludge aeration basin. So the aeration word means that it is the air molecules are circulated within this sludge or the waste water. So this inlet is the inlet, the wastewater comes in with or without primary clarification. Primary clarification basically just means removal of solid particles. So in the wastewater that is usually generated in the urban settings, there's also active solid particles. So sometimes the solid particles are removed, or it's sent into the aeration basin without removal of those particles. And then this goes into a clarifier where all the solid particles are settled in the bottom, and the clarified water comes on the top. And after that, this clarified wastewater is just sent out as an effluent containing a lot of components still dissolved in it. These are some of the parameters that they measure: the total solid content that is dissolved in the water, the biological oxygen demand. Biological oxygen demand just means the amount of organic matter that is there in this water that can keep consuming oxygen. And these are some of the values that are for a conventional wastewater discharge. Okay, there are more people. Yeah. Yes, let me just go back and yes. So conventional wastewater treatment does not account for the nitrogen content in the treated water. So here we can still see that there's dissolved nitrogen content, but that's usually not taken into account. High nitrogen content results in eutrophication and algal blooms. This is one of the problems that is commonly seen in many of our local water bodies. That is, algae starts growing in such high amounts. what is the problem of algal blooms is that these start growing on the top surface of the water, thereby preventing the reach of sunlight to organisms that live inside the water. So the algae occupies the surface of the water completely and no sunlight reaches the bottom, and this is a major problem for aquatic life that depend on the sunlight reaching them. And these algae also continuously consume oxygen, thereby reducing the overall oxygen content. So this high nitrogen content actually serves as a substrate for them to grow, and resulting in production of high amounts of this algae. Algal blooms or increased oxygen presence reduces dissolved oxygen content. So this not just prevents the sunlight from entering the water, but also reduces the dissolved oxygen content in the water for the aquatic life. So biological oxygen demand of the wastewater that does not take into account the dissolved nitrogen content increases because of the algal bloom, and thereby reduces the dissolved oxygen content. So what is a remedy for this? How do we use better techniques? Is by doing biological nutrient removal. So this, along with the conventional method of removing the solid sludge and aeration zone, also involves a couple of other zones, which is anoxic or anaerobic zone, where the water is deprived of oxygen, and then there's an anoxic zone where the N2 is allowed to release, and then there is a conventional aerobic zone. So in this process, when after the N2 gets released from the so here in this zone the microbes grow, so the microbes are added in this zone, and then they result in the removal of nitrogen, and then this in the end results in conversion of ammonia like products to nitrates, which can be easily removed from this process. So this is an intern there's also an internal recycle where they keep removing the nitrates and then giving it back to this zone, so that the bacteria that are grown in this step can use this nitrates for removal of nitrogen. So they convert the nitrates and result in the escape of nitrogen. So in this the end of this process, the biological oxygen demand also significantly reduces if we compare to the previous step, which where it was around 20, it has become 10 milligrams per liters, and the total nitrogen total okay, yes. Okay, there are also couple. I think there's also another question. Okay, Prarthana, I'll come back. Okay, this question is about superbug. Okay, let's just go over this. Um, Superbugs, which are more efficient in degrading hydrocarbons or other pollutants, must have one of the following: high survival in the presence of different pollutants, ability to express genes for degrading single pollutant, high growth in spite of low production of degradative enzymes, low survival rate in contaminated environment. Okay. Uh, so, if this is a question from this week's assignment, I'll not be directly able to tell you the answers, but we can always discuss one by one some of the things that are there. So, for the others, I'm just reading out this question that Prarthana Kaushik has posted on the chat box. So, this is about the superbugs. So, we saw about superbugs in this previous slide. Maybe I'll just keep this slide on. So this says high survival rate in the presence of different pollutants. So these are some of the things that I explained. So I explained about stress tolerance, how much stress it can withstand. So high survival rate in presence of different pollutants would mean the spectrum. So they are asking which are most efficient in, so superbugs which are most efficient in degrading hydrocarbons or other pollutions must have which of the following characteristics. So we have to choose one of the characteristics that's the most important. So is it the is it about its survival rate in presence of different pollutants, whether it is the spectrum that determines the efficiency, is it the ability to express genes for degrading a single pollutant. So this means this has nothing to do with the spectrum. So here it just says that it should have genes to degrade a single pollutant, but we would call anything more efficient if it has a huge spectrum and it can work on different pollutants given its genetic makeup. Higher growth rate in spite of low production of degradative enzymes. And so if the degradative enzymes that it is producing is low and there's just higher growth, then it means that it has a higher growth rate. But what we want is that it should produce enough degradative enzymes so that we can use it effectively for degrading oil spill or any wastewater or any management objective of our interest. So, do we really want it to just keep growing, or do we also want it to produce enough degradative enzymes so that we can use it for our requirements? Low survival rate in contaminated environment. So this is just the exact opposite of option A, right? So if it is not going to survive in an environment with lot of pollutants, how are we going to use it for anything of our need? So these are the 4 points. So I hope you guys understood what I was trying to explain about each of the points, and you will be able to select the right option about the superbug. So basically, just recapping, our superbug should be something that is effective against multiple pollutants, has a huge spectrum, has a very high efficiency. It should be able to withstand or tolerate high stress conditions and should keep degrading organic molecules for a longer time with lesser amount or lesser production as well. So couldn't see the slide. Yeah, sorry, I'm not presenting anything currently. So okay, thanks Prarthana, I hope that clarified. Yeah, so now I'm sharing again. So we were here. So can you guys see the screen again? Can someone just confirm if we are back on track? Can someone just say yes?
Yes, ma'am.
Yeah, okay, thank you. So I hope that helped. So if there is something like this, a specific question about something that we are going over now, please stop me in between. You can unmute and just say, please take a look at the comment box or whatever, so that I can stop sharing, because I don't really see the comment box when I'm presenting my screen. Okay, so coming back to this, we were talking about using microorganisms in wastewater management. So there's an anoxic zone or an anaerobic zone where the microorganisms grow, and then in the next zone, they are allowed to convert nitrates into nitrogen, and this is like a cycle because in the next step, the ammonia is converted to nitrate once all the nitrogen is removed. Okay, there's someone else again waiting. All right. So that's biological nutrient removal, which involves using anaerobic bacteria in an anoxic chamber for removal of nitrogen. Distinct bacteria are chosen to remove ammonia, nitrates, and phosphates. So, as you can see here, the total dissolved solid content reduces, the total nitrogen reduces, the total phosphorus content also reduces. So, for each of these conversion, we will use a different type of bacteria that can chew up ammonia, that can chew up nitrates, that can chew up phosphates, and in the end, we will have water that is completely ridden of these external nutrients, which are a major problem to our natural systems. So the waste sludge that is collected in the bottom goes for decomposing or composting, where it can be used as manure, and the clarified water is discharged into the water bodies, but now with lower biological oxygen demand or lower nitrogen content. Understanding the ecology of the microbe helps in this selectivity. So here I said that we will use different types of microbes for converting nitrates to nitrogen, ammonia, or to degrade phosphates. So to know what microbe actually converts each of these things, we need to understand what specifically they convert. So there, I'll again just come back and just read out from the comment box. So this is another question that Prarthana had posted in the beginning of the session. So here it says that while using mathematical modeling for integrating a large number of microbial community of environmental significance. So here, like I said, in this wastewater sludge, we will be using different microbial organisms for phosphates, nitrates, and so on. So how much of each would you use? So that's when it is important to make a mathematical model which will accurately calculate the amount of microbe that, or the amount of growth that we would need to produce the exact number of products in the output for a given amount of wastewater. So all of this is what is taken into account. So which is the most essential system component that must be identified by a mathematician? So, critical type of microorganisms, substrate consumed or utilized by the microorganism, the product formed by the microorganism. So this is a question for us to think about. So, these are all the different things that are going to be in the equation: the substrate consumed or utilized, the product formed. All of these things are different components that are there in the part of the equation. But what is the most essential component? I'm not going to directly give away the answer, but this is something that's there in this week's assignment, so I want you guys to go back and think. But let's just imagine this wastewater example. So if you were making this calculation, what would you think about solving first? Do you want to know what kind of microorganisms to use? But if you already know what kind of microorganism you're using, then you would also know what kind of substrates that given microorganism is consuming, or the kind of product it is producing. So what are some of the things that you would take into account if you're choosing a microorganism? So that, I think, gives away the answer of what would probably you be taking into account when you're trying to use one of these microorganisms in your wastewater treatment plant or in any of the other applications. So yeah, let's just go back to this slide. So far we saw that protecting our water resources and development of alternate energy are areas that need environmental biotechnology innovations. And any country which is looking to address some of these problems should be investing in some of these environmental biotechnology innovations. And microbes could do both, because they don't just degrade the waste products, but they can also use those waste products as energy and develop, and that can be used for developing an alternative energy source. All right. And studying microbial ecology helps us in understanding the niche, the abundance, diversity of microbes in an environment, the enzyme produced, the substrate leave, the byproducts of microbial action. So this is a little bit of recap from our previous session as well. So understanding microbial ecology is very important, because you need to choose the kind of microbe that you want for a particular reaction, let's say wastewater treatment. You need to know what are all the different microbes it can interact with and effectively stay in your wastewater treatment plant. You need to know what are all the different actions that it performs in the natural habitat in its niche, so that you can utilize some of those for your own applications as well. What is the general abundance? Knowing the abundance will help you in determining what is the critical number of microbial growth that you need to achieve in a particular environment, because there is something called contact inhibition that happens in a lot of cells. If there is an excess of microbial growth and excess consumption of resources, some of them will start dying because they are now not getting enough nutrients. There's excess of growth. So if even if you have seen like a typical microbial growth curve that we do in our undergraduate microbiology sessions, there will be a dormant phase where the microbes will just be picking up. They'll be just utilizing resources, but they'll not be growing. But after that phase, they start growing with an exponential rate. And then there'll be a phase where they will saturate, or they'll become like a flatlining of growth happens, and then after some point the growth will start deteriorating, which means that they'll start dying because now there are too much of them and there's not enough substrate to utilize. So that is also why we should know their natural abundance, so that we can give the right amount of resources and the right amount of time for them to grow. So let's say, for example, in a wastewater treatment plant, if they are growing in a particular tank where there's wastewater, you need to know the exact amount of time for which you need to provide an anaerobic condition and when to switch to an aerobic condition, so that they don't start deteriorating because of the lack of resources. And diversity of microbes, the enzymes that they produce, which substrate that they can effectively use for producing our product of interest, and what are all the byproducts that can either be used as an alternate source of energy, or it can be a biopolymer that they produce during these processes. So that's pretty much is what microbial ecology encompasses, and each of this would have been dealt in detail in the original lectures, so I can't really go over each of them in detail. But in any of these lecture slides, if you had any questions, you can always post them to understand more about the applications of microbial ecology. And the next thing is to know a little bit more about the microbial metabolism itself. So we spoke about choosing substrates, we spoke about what happens to the substrates, but how do we in practical actually understand what kind of substrates are cleaved by microorganisms and so on. So for before going to that, we need to understand this basic simple two processes that happen not just in microbes but in our body, plants, and every organism that uses some form of energy is constantly undergoing this process of catabolism and anabolism, which is together called as metabolism. So, catabolism is a process where larger molecules are broken down into smaller molecules, which also produces energy in the process. Let's say we consume food. So, these are larger molecules or building blocks which are broken down by our body to produce the energy. So, any food that we consume is what gives us energy. So, the carbohydrates, which are constituted in most of our food, let's say rice or roti or anything, is carbohydrate rich. And when they are broken down, they are basically complex molecules of glucose, and that is converted into energy, that is adenosine triphosphate or ATP, which we have learned is the energy currency of the body. So catabolism is the process that actually breaks down this complex carbohydrates to produce energy molecules. And what is anabolism? Anabolism is now exactly the opposite of that. Smaller molecules are assembled to produce larger molecule, and this uses energy. Plants produce food. They do photosynthesis by utilizing sun's energy, and they take in the carbon dioxide from the atmosphere. And these carbon dioxide are the actual building blocks. They also take in water, so they use the carbon and hydrogen in the water and the air to make this complex carbohydrate. So carbohydrates are basically carbons plus hydrogens. Hydrates are hydrogen along with water. So this is what makes complex carbohydrates. So plants actually take the sun's energy and they fix the sun's energy to combine these smaller carbon and hydrogen molecules to make complex carbohydrates. And this is a process that uses energy. So this is catabolism and anabolism. Anabolism also happens in our body. That is how fats get stored up in our body. So any excess glucose that gets broken down in the body and that's not being used for production of energy will now be converted into fats and stored in our body for future use. So that is a process of anabolism where smaller molecules are assembled to produce something complex like a fat. So this catabolism and anabolism continuously keeps happening in plants, microbes, and all living organisms. So why is this important for understanding about microbes? Because this is where we can use microbes effectively, because microbes can utilize a variety of substrates, and produce a variety of products. So in the example that I gave, carbohydrates was the substrate and the product was glucose and energy. But in the same time, microbes can use a wide range of energy sources, including biomethane, bioethanol, biohydrogen, as energy carriers, and they can produce a wide range of substrate along with production of energy. So some of these things, such as rock weathering, or processes that keep happening in the nature. So this is like a huge combination of different kinds of products that are produced from different substrate reactions. So these are some of the complex molecules that are converted by microbes. So fermentation is a very common reaction that happens in our day-to-day life. Dosa batter is fermented by microbes, and that's how we get dosa. So any batter which is a complex carbohydrate, a starch, is converted into a different product with the help and also with the production of carbon dioxide. So this fermentation produces acetate, butyrate, propanate, and hydrogen, and different kind of molecules. So these are some of the common examples that we say in our day-to-day life, where microbes act on complex substrates for production of different organic compounds. This is a summary reaction. The main take home from here is products such as biomethane, bioethanol, biohydrogen can also be used as energy carriers by microbes for production of energy, and also for degradation of harmful substrates. But how do we determine the substrate utilization of the microbe? So how do we know which substrate consumes what microbe? Okay, there's someone waiting. Okay, let me just go back. Yeah, how do we determine which substrate is utilized by a microbe? So there are different omics techniques that have evolved throughout the technological advances that help us in studying different things. So we also briefly saw about these omics when we were trying to understand how do you find out the species of a microorganism. So some similar techniques can also be used in order to understand the substrate and the microbe relationship. Transcriptomics allows us to study the mRNA and gene expression, and based on this gene expression, we can know what kind of genes are produced by the microbes, and those genes in turn produce what kind of enzymes, and hence we can understand the substrate that can be cleaved by that particular enzyme. Proteomics is also very much related. Here, you directly sequence the proteins or the enzymes that the microbe produces, and also understand about the activity of these enzymes, so that we can correlate that with the substrate that it utilizes. And directly understanding the metabolic intermediates produced by the microbial organisms through metabolomics. So this chart kind of summarizes all of the different techniques that are involved in identifying the substrate that are utilized by microbes. Some of these are in vivo and in vitro techniques. In vivo meaning you directly look at inside the microbial organism what is happening, and in vitro techniques are you basically extract their transcriptome, proteome, and then understand what is happening. So stable isotope microbiomics. So this is an in vivo technique where you can use the whole extract or a fraction to understand what kind of metabolites are being utilized. So IRMS is isotope ratio mass spectrometry. So mass spectrometry basically is a method of understanding the mass composition of the different ions that are present in any particular compound. Depending on the ionization of the particular compound, they ionize at different ratios, and then we can find out what is the overall mass of the particular compound that we are ionizing. So there is an ionization chamber. The molecules ionize depending on the different atoms that are present, the different ions that are present. Uh, okay, yeah, they ionize depending on the different ions that are present, and then once they ionize, just a second, yeah, once they ionize, we will you put them through a detector, and then the detector detects the different ions that came from a particular molecule. So that's basically any how any mass spectrometry works. And in case of IRMS, it's the isotopes that gets ionized, and based upon the different stable isotopes that are present in a given particular molecule, we will know what are all the substrates that have been utilized by the microorganisms. And there are also these different methods using RNA, which includes quantitative PCR to understand the different genes that are synthesized by the microbe, microarray techniques, RNA transcriptomic techniques, protein techniques, and this can also be done at a single cell level. So we don't have to have lot of microbial product to actually understand the substrate. So we can even do this at a single microbial cell level to understand what is happening. And these are also some of the techniques that are in vitro techniques that we can do after isolating the products. So these are directly done on the live microorganisms in under in vivo condition, and these are done on the products of the microbial organisms. So how or what is the base product for some of these different techniques? So the natural organic matter composition can be analyzed by some of these techniques, such as IRMS and NMR, which I had explained earlier also, IRMS. So these are some of the techniques that we can do on natural organic matter. And nano sims and technologies like that are done on mineralogy. So here these are different zones. So vadose zone is the unsaturated zone, and variable zone is it's the variable saturation. So this saturation basically means the soil that is there and the underwater water table, and how much of this water is completely saturated. So this saturated zone is completely saturated, so the soil is completely saturated with water. So this is an 100% saturated water zone. So these are the different zones that exist between the soil and the underwater water table, and microbes exist in these different layers depending upon their water requirements. And by isolating microbes from each of these zones and performing some of these techniques, we will be able to understand the substrate that they consume under each of these different conditions. So yeah, so this is an overview of how nano sims work. So this is stable isotope mass spectrometry. This can be done at a very nano level, so that's why it's called nano sims. To understand what kind of substrates are utilized by microbes, this is a very effective technique, because we can individually label different substrates using fish. So fish is a technique which abbreviates as fluorescence in situ hybridization. So we can individually label different substrates with different stable isotopes. So stable isotopes like different isotopes of carbon, different isotopes of oxygen, and all that exists, and we can differentially label each of these. Usually carbon is what is used in many of these stable isotope methods, so we can label each of these substrates with different isotope of carbon. And once that labeling is done, we can put it through the stable isotope mass spectrometry, and it analyzes. So we will incubate this differentially labeled substrate with the microbes. And fish helps us in understanding by just visualizing using fluorescence. So if a particular substrate that is labeled with a particular radioisotope and then with a particular fluorescent molecule is cleaved by the microbe, we can visualize this under in Site2 conditions, whichever substrate is being cleaved by the respective microbe, we'll immediately get the output. And this can also be sequenced to further understand what exactly in the substrate is getting cleaved. That is where we will use sims. So we will specifically separate out depending on the output that we get from fish, and then we will subject it through nanosims, and we can individually label like this nitrogen isotopes or carbon isotopes, and we can see that here C13 labeled substrate is what was getting cleaved. So we can sequence each of these substrate individually, and we will see that if a particular substrate was cleaved, this will be broken down into two different molecules, and using mass spectrometry we can identify the differences in the masses of the substrate that were cleaved, and we can say that okay, this substrate that was particularly labeled, let's say bioethane, is what is used by this microbe for generating energy, and therefore if we use this particular substrate in our, or if this substrate is there in the waste that we want to treat, we can use the respective bacteria. More about this idea of nanosims itself is available in this particular paper where I've got this reference from, so you can always go back and check what is happening here. So this is how the mass spectrometer in general works, and this is how nanosims also work. So there is a primary ion source which ionizes the sample, and once the samples ionize, they pass through an electrostatic sector and a magnet through which they get deflected towards different detectors, and depending on what is their mass after either cleavage or without cleavage, they'll get detected at different elution rates, and then we will know that from this acquisition we will know what exactly is the mass of those individual components that got ionized. So that's pretty much about it that I have for today. So if you guys have any specific questions related to the assignments, or if there's anything that is not clear in these slides, I can always go back and explain. So now it's just time for questions. Yeah, any other questions from the assignments or from the lecture slides? Anything, if it's not clear from the presentation, you can always ask.
Ma'am, what is aquastation and treatment?
Sorry?
Ma'am, what is aquastation and treatment that you said just now?
Yeah, so acquisition is the acquisition of the ion pattern data itself. Treatment would either mean you further subject them to further treatment, or like subject them to smaller portions so that you can detect them at a different level. So acquisition basically is what is the first step of getting the data from the detector. So once the ionized molecules get deflected by the magnets, they get detected, and the data is acquired by the detector. So that's the acquisition. Any other questions? Okay. Anything else, guys, from the previous slides or from the lectures that are not clear? I hope you've been able to solve the assignment questions so far. So if you have any doubts, we can always discuss them. Any questions, guys? I'm hoping it was all clear. And all of you are completing your assignments, right? So in case anyone has any difficulties finding out where those assignments are or solving those assignments, please do let me know. People are still joining, but whoever just joined, the session starts at 6 every Tuesday, and I usually end it around 6:50 with a spare time of 10 minutes in case people want to ask questions or clarify doubts. So the session is between 6 to 7, and the session is recorded, so you can always find the full recording of the session on YouTube. In case you have any doubts, you can always go back and check the recorded sessions. Um, yeah, I hope I answered Prarthana's questions as well. Prarthana, if there are other questions that you're not sure about, you can post them too. Thank you. It's my pleasure for answering the questions. That's why I'm here. So I would expect it better to be a discussion session, like you guys post questions and then I answer, rather than me just presenting a couple of slides, because this should be a live interactive session. Is there anything specific that you guys want me to put in the lecture slides so that it would be of better help to you all? All right. I only see a couple of names from the previous session. Many of you are new, I suppose. Okay, in any case, if there are no more questions or clarifications, I can close the session here. I hope all of you are understanding whatever we discuss here. In case it's difficult to follow, please do write to me. If you want this session partially in regional languages, you can ask me. I can try. So you will have my mail ID. You can always mail to me through the discussion forum. Yeah, in Hindi. Okay. My Hindi is not really that great, but I can understand. If you have questions in Hindi, you can always ask them. You can unmute and ask them or type them. I just have difficulty speaking Hindi. My Hindi is not that great. Uh, but you can always post your questions and I can try attempting to answer them um, as much as I can. But yeah, sorry about that. But I think the NPTEL team is putting in efforts in organizing regional sessions. So um, hopefully soon you'll have better regional sessions. All right, if there are no more questions, thank you so much everyone for joining. I'll save the recording and post it on YouTube. You guys can watch it there as well. I hope these sessions are of help to you. Thank you so much for joining.
Thank you, ma'am.
Thank you.
Thank you, ma'am.